Hello friends, again welcome to Ultimate Education. I am Farooqsir here again with the presentation of Sexual Reproduction in Flowering Plants Part 1. The flowering plants show sexual mode of reproduction and it have complex reproductive units as male and female reproductive units along with the accessory structures. The flower actually is a modified stem which functions as a reproductive organ and produces ova and pollen grains. A typical angiospermic flower consists of four whorls of floral appendages which are attached on the receptacle, which you can see from the figure which is given over here. The calyx, the appendages which are attached to the receptacles are calyx, which is called sepal, corolla, which is also called petal, and the reproductive organs like androecium, that is stamen, which consists anther and filament, and then gynoecium, carpel, that is the female reproductive structure which is composed of ovary, style and stigma. Now we discuss the different stages in sexual reproduction in flowering plants. The first stage is pre-fertilization stage. Here we are going to discuss and study the structure and the other events which take place in sexual reproduction. The flowers, it consists male reproductive structure which is called androecium and the female reproductive structure which is called gynoecium, as I told you in my previous slide. The male structure and female structure, they consist gametes, right? Both male and female gametes are produced. Androecium consists stamen, microsporangium and pollen grains. The stamen consists of long and slender stalk-like structure which is called filament, and generally it is bilobed anthers as you can see in the diagram. Each lobe contains two theca. Theca means that is diathesis, two theca layers, right? The anther is four-sided structure which consists four microsporangia, two in each lobes. Microsporangia develop further and become pollen sacs which contain pollen grains. The second unit of male reproductive organs, that is microsporangia. It is generally surrounded by four layered walls. First wall, epidermis; second, endothecium; third, middle layer, and innermost tapetum. Tapetum nourishes the developing pollen grains. Sporongenous tissues, it is compactly arranged of homogeneous cells which are present at center of each microsporangium when the anther is young. Microsporogenesis, this process is the formation and differentiation of microspores, that is pollen grains, from microspore mother cells, which is also known as M.Sc in short. By reduction division is called microsporogenesis. The cells of sporongenous tissues undergo meiotic division to form microspore tetrad. As the anther mature and dehydrate, the microspore dissociate and develops into pollen grain. Now about the pollen grain. The pollen grain represent the male gametophytes, the main responsible gametes which is responsible for the fertilization process. The pollen grains are made of two-layered wall. First, axine. It is made of sporopollenin, which is most resistant organic matter. It can withstand high temperature and strong acid and alkali. No enzyme can degrade sporopollenin. Why it is so? Because this is the main responsible male gamete which helps in fertilization. If it is weak, then any high temperature or strong acid or alkali or any other matter can destroy it. That's why the nature has provided it the security. And second one, intine. This is the thin and continuous layer made of cellulose and pectin. Then the other structure on the pollen grain, it is germ pores. It is a small aperture which is situated on axine where sporopollenin is absent. It forms pollen tube which helps in fertilization, and that pollen tube reaches up to the ova of the female structure. A plasma membrane surrounds cytoplasm of pollen grains. A mature pollen grain consists of two cells. They are called vegetative cell and generative cell. Both are with the nucleus. The vegetative cell, they are bigger in size. They have abundant food reserve. They have large but irregular nucleus, and they are responsible for the development of pollen grain. While the generative cell, which is the main cell which take part in the fertilization, they are small in size. They involve themselves in syngamy, that is fusing with an egg, and they are having dense cytoplasm and nucleus. The viability of pollen grain varies from species to species and should land on stigma before the period to germinate. Nowadays, the pollen grains of large number of species are also stored in liquid nitrogen at temperature minus 196 degree at pollen bank. Now, the female reproductive unit, which consists the pistil, the megasporangium, that is ovule, and the embryo sac. The gynoecium consists of single pistil. This position is called monocarpillary. The flower which has single pistil is called monocarpillary, or it can be more than one pistil. That position is called polycarpillary. It may be fused position, that is called syncarpus, or each pistil can be free. That position is called apocarpus. The example of multicarpillary along with the syncarpus pistil is papaver flower, and the multicarpillary and apocarpus pistil, that flower example is michelia. Each pistil has three parts: the stigma, the style, and the ovary. Inside the ovary is ovarian cavity, that is called locule. The placenta is located inside the ovarian cavity, and the megasporium ovule arise from placenta. Now the other thing, megasporangium, which is called ovule. Let us discuss about the structure of the ovule. Ovule is a small structure which is attached to placenta. It has many parts. First part, that is called funicle. Funicle, it is a stalk-like structure by which the ovule is attached to the placenta. After that, at the hilum part, that part is the junction between the ovule and funicle. It has got two integuments, that is inner integument and outer integuments. They protect the egg, they protect the ovule, right? Another part is micropyle. Micropyle is a part which it is a small opening at the tip of ovule through which the pollen tube enters into the ova, right? And then the fertilization takes place. Then the another part is calaza. Calaza is the basal part of ovule, and it has another part that is called nucellus. It is 2n, they have got mass of cells. They are enclosed in integuments. They have got abundant food reserves. The process of formation of megaspore from megaspore mother cell by meiotic division is known as megasporogenesis. This process takes place in ovule. Ovule differentiates a single megaspore mother cell, which is also known as M.Sc in short, in the micropylar region of nucellus. M.Sc undergoes meiotic division that results into the production of 4 megaspores. In most of the flowering plants, 3 megaspores degenerate and only 1 megaspore develops into the female gametophyte that is called embryo sac. The nucleus of functional megaspore undergoes first mitotic division to form 2 nuclei which move to opposite poles to form 2 nucleate embryo sac. 2 more sequential mitotic division take place, which results into 8 nucleate embryo sacs. Out of these, the 6 of the 8 nuclei surrounded by cell wall and remaining 2 nuclei which are situated at polar nuclei, which are also called polar nuclei, are situated below the egg apparatus. 3 cells are grouped at micropylar end to constitute egg apparatus and 3 cells at callosal end forms antipodal cells. At maturity, the embryo sac is having 8 nucleate and it is 8 cells. Thank you friends for watching our first part of sexual reproduction in flowering plants. The second part will follow very shortly, and we request that if you like our video, please like it, share it and subscribe it. Thank you. See you very soon.